It's a real pleasure, although a sorrow too, to introduce Bill Reeburgh, who's going to be retiring at the end of the month, I guess. For those of you, we've brought back some old students and old faculty members. For this talk. I don't want to take much time, former. I don't want to take much time away from Bill, but I thought it would be just appropriate to say a few words of introduction. I mean, he got his bachelor's degree in chemistry from the University of Oklahoma. He did a PhD and a master's degree at Johns Hopkins in oceanography. And but for us at UCI, really, Bill's career begins when he comes here from the University of Alaska at Fairbanks. I think those of you who are familiar with ESS, and everybody is here in part because of the department, Bill is really one of the founding faculty members, but I think his stamp is more imprinted on the department than probably any of the rest of us. Bill arrived here, I think, officially in 1993, but I remember talking to him on the phone about whether or not we would come and what kinds of stuff we would ask for our labs and what kind of work we might do if we come here. Bill pretty much was the person who pushed through the proposals that became the, allowed us to grant PhDs in the department, and then at that point, we were the program in geosciences. He pretty much wrote and helped push through the PhD program and the department proposal, which were two separate things, and they both got approved, I think, in kind of record time, given that they have to go all the way up through the state legislature. At that point, we became the Department of Earth System Science, and it's been, I think, a big success. It was an experiment at the beginning when we all got here. Bill also served as the chair in a very important period in the department from 2000 to 2003. Pretty much his chairship ended when we moved into Crowell Hall here. And so, he was instrumental in getting that. There are about 5 or 6 or 7 faculty members that Bill was instrumental in hiring. And so we are, in a large part, the department we are today because of Bill Reeburgh. So, and he's asked to, he's giving the seminar today, I think, to talk mostly to students about a scientific career. I do want to say we're hoping to have a special session for the science part of Bill's career at the 2010 Ocean Sciences meeting. So we'll have a special session there. It'll be in Portland, so in 2010. All right, so without more, thank you, Bill.
Thank you. Well, Sue's right. As of sunrise tomorrow, I become Professor Emeritus, and I separate from UCI on the next day, and then I'm obligated to be around till the end of the quarter. So our plans after that are to move to Vancouver, Washington, so we can be closer to 2 of our children. And we'll pick up and resume from there. Sue mentioned a bunch of people who've come today, and I wanted to just point out how I'm happy I am to have them here. Jerry Schubel and his wife Margaret, probably the oldest friends in the room. Jerry's the president of the Long Beach Aquarium, and he was my best man. So in fact, when Carolyn would come down to visit as we were recording, they would house her, they would keep her. They were the chaperones. So we go back a long, long way. Our daughter Nancy is here, Nancy Bacon, and it turns out that guys like me are incredibly fortunate to have 2 families, a real family and an academic family. The academic students family that all UCI graduates, the ones I see here, Jennifer King, Dave Valentine is back there, and John Kessler, there he is, right in front, okay. And Mary Pack, who is a current family member. So anyway, I'm glad that all of you are here. What I wanted to do today was to direct my remarks mostly to students and postdocs, because as I talk to people, I sense that everybody's looking for the magic arrow, the formula, the golden button. And I want to assure you that no such thing exists, that there is no such thing. And so what I'd like to do is to take you on a journey through methane biogeochemistry. So let's start off with that. Come on, back. Okay, here we go. Okay, so anyway, this is what I'm going to do. A lot of this is based on a talk that I gave. I felt it was a fairly effective way of covering this material. I gave a talk about 3 years ago at the International Union for Microbiological Societies, where I talked about this anaerobic oxidation, and it was a very controversial process. I think I ran into more opposition than anything for about a 10-year period, and it was a real education for me, but I survived it, and I feel I'm stronger for it, and I'm just would like to share some of the reactions that I had. What I'm going to do then is to take you through this. This diagram here has sediments and water column, and it has a whole series of topics, and I want to emphasize that these are all, you're benefiting from my 2020 hindsight. So every one of these steps, these milestones, I view a milestone as being a point where you have enough data, you understand things well enough to where you can ask the next question and formulate the next piece of research. And so each one of these goes on, and the point is that when I first started this, I couldn't have anticipated any one of the future ones. So every one of these was something that came from looking at the data and figuring out where you stood. And a lot of it was done in sediments and the water column. And so what I'm going to try to do is to step through these. I'm not sure I'll make it all the way through, so I've got a point where I can break after I cover global importance and do other things if we're taking too much time. The other thing I have to do, sort of required, is a methane budget. And this is one that we put together, and what I want to emphasize here is that most of the atmospheric people look at this one here, the net emission budget, and this is where methane comes from various sources, enters the atmosphere, and you can see down here at the very bottom, most of it is consumed in the atmosphere by photochemical processes. A little bit is taken up by soils, and that's sort of the thing. What I've emphasized all my career has been consumption, and in particular, I've worked in the ocean, and I've also worked on wetlands, and it turns out that these are unique because these two zones or regions are set up so that there are bacteria that live there that oxidize the methane, and they essentially stop the flux to the atmosphere before it happens. So there's a whopping amount of methane that's released but never sees the atmosphere because it's oxidized by bacteria. Okay, the next thing I want to do is emphasize too that you won't hear me say, I'll talk budgets, but I won't talk methane cycle, because I don't think there is a methane cycle. I think that what happens is the fate of methane is to be oxidized, and that's it. It basically just shoots through, gets turned into CO2 in the atmosphere. Okay, the next thing I wanted to go to was sort of the approaches that geochemists take, and I've always sort of relied on these two approaches to help out or help me keep, when you're swatting mosquitoes at Toolik Lake or something like that. These always help. The 4 R's from Dayton Carrott. Dayton Carrott was an early faculty member at Johns Hopkins, one of the co-founders or early founders of CBI, the Chesapeake Bay Institute. He always used to talk about, it's easy, all there are is the 4 R's, roots, rates, reactions, and reservoirs. And it turns out that these aren't in any particular order, but you need to know something about reservoirs and rates in order to assemble budgets and go this way here. And Wally Broker does it in a different way. Wally refers to what he calls inverse chemical engineering. Wally maintains that the Earth is a finely tuned chemical plant that's working, and we don't know the blueprint. We don't have an instruction manual, and that it's our task to size the pipes, measure the fluxes, and figure out how the whole system works. So this is kind of the role of what geochemists do. Now, the next thing I wanted to do is to move on down this thing. So these milestone posters will come up periodically. I'd like to talk about methane concentration profiles. And the first thing I want to deal with is a squeezer. It turns out that I had worked for a master's degree at Johns Hopkins. I'd done a very meticulous, high-precision study of electrical conductivity of seawater. I spent hours and hours following a galvanometer, the ball on a galvanometer, and I really wanted to get into the field. So one of the first things that we did was actually Jerry Schubel and I were formulating our PhD proposals together, and I put together the squeezer. Interstitial water in the 70s was a big deal. The deep sea drilling project or ocean drilling program was just starting, and people were looking for ways to get the fluid out of sediments. And it was a system where you had high surface area, small volume, and we expected things to sort of stay put, that they weren't going to be just swept away by currents as they are in the ocean. So I made this thing here. It's made of, the previous versions were made by Raymond Seaver at Harvard, and they were a big stainless steel thing that you filled with mud, and it had a piston and a big crank that you turned. It was bolted to the table, and it jammed, and it had to be taken back to the machine shop to unjam. And so I decided to make something simple, and what we have here is a container here in the center. That's where the sediment, the mud goes. There's a set of filters at the bottom, and this number 4, that piece is a piece of rubber sheeting, dental dam rubber, and the whole thing gets clamped together, and you use gas pressure to do this. So anyway, once I had the squeezer and was convinced it worked, we were supposed to write PhD proposals, so a 1 page. So I wrote 1 page where I was going to study gases, CO2 system, metals, nutrients. It was incredibly ambitious. It was about 10 careers, and I ended up selecting gases because it looked like that was where the most action was. So the next slide, then we go to how we did that. So what I used then was a squeezer, and I squeezed water, pour water, interstitial water out of the sediment into this device. This is what's called a sampler stripper. It's got a little frit here, and it turns out that we could measure the volume of a discrete sample and turn it upside down, mount it on a gas chromatograph, and analyze the gases. And some of the results we got at first were puzzling, but at the same time kind of challenging. It turns out that methane, the M's, and I refused to connect the dots. I figured that these are discrete samples, and so I at that point just did this way. But methane is concave up. In other words, you can see that they go like this in both stations. These are 2 stations of different depths. And other strange things happened with nitrogen and argon. I measured argon because it was an inert gas. It did no biology. And it turns out that instead what happened with the argon was that it equilibrated with methane bubbles and was removed from the system. So it wasn't static, it wasn't conservative. And it turns out that we came up with this. Well, the concave up distribution was puzzling. And so we started wondering what could cause this. And so one of the things that I thought was, I figured there must be, the sediment's flocculent, there must be some oxygen being mixed in. That was all I could consider. And so we get into this whole question about the involvement of sulfate. Well, I moved to Alaska, had an opportunity to work on lakes. I essentially reestablished the system and got it going, and we worked on several lakes. And one of my students there, David Heggie, and I worked on this, and we realized that there's a difference in the general profile between freshwater and marine systems. And it meant that since the major really reactive constituent of seawater had to be sulfate. So I figured, okay, must be something there, but I wasn't really convinced. About the same time, Chris Martins and Bob Berner at Yale, Chris was a postdoc with Berner at the time, started doing jar experiments. And these are very simple things. What they did was to get a big bucket of mud from Long Island Sound. They filled mason jars with the mud, and they closed them up and let them incubate. And so this is a time on this axis here, and you can see that methane doesn't begin to show up and really be produced until all the sulfate, or almost all the sulfate, has disappeared. And so they were doing diagenetic models and trying to figure out what was happening. And this I thought was all very interesting. It sort of fit in with my general ideas. And then I had an opportunity. We were doing diagenetic models and advection diffusion models. And this basically sort of is the sort of guide slide for advection diffusion and steady state models, steady state advection diffusion and diagenetic models. It turns out they're all based on the same equation, this 3-dimensional distribution of variables equation, where we have the rate of change, rate of change of concentration with time. We have diffusive terms in the x, y, and z direction. We have advective terms in the x, y, and z direction. This J in Harmon Craig's notation is the reaction term, and then this was the radioactive decay term. And this was the equation that was basically sort of served as a centerpiece of the geosex program. Okay, well, in Berner's case, Berner wrote a book about diagenesis, and he simplified things to where he basically made it one-dimensional. And the difference between these two is that in the case of diagenetic models, the diffusion coefficient, the A or the D, is a tortuosity-corrected molecular diffusion coefficient. So if you consider that they're particles and water together, the ions, which are in the liquid, have to migrate around particles, and so it turns out that tortuosity-corrected diffusion coefficient is about a factor of 100 smaller than a regular molecular diffusion coefficient. And then W, since the sediments are basically accumulating particles from the top and the interface is rising, it turns out that it's the sedimentation rate. So this is sort of what we we realized was important. In the case of advection diffusion models, A is an eddy diffusion coefficient, and these are typically on the order of 10 to the 5th centimeters squared. They're a factor of 10 to the 10th bigger than the molecular diffusion. And this W is the vertical advective velocity. And the only place you can get a vertical advective velocity is in a basin with a sill, like the Black Sea or the Cariaco Basin. So these were the first times these really were used and really worked. Okay, well, we went to the Cariaco Basin. I had an opportunity to go with Kent Fanning and Michael Pilson. In 1974, we were on the Trident, and which was a ship that they got before the one they have now. And we worked at 2 stations, one in each of these 2 basins in the Cariaco Basin. This is off the coast of Venezuela. And we basically made measurements. I measured methane in sediments. Dennis Wiesenberg made methane measurements in the water column. He took the samples and shipped them home. And one curious thing is that John Kessler and I were in the Cariaco Basin 30 years to the day after we left it. We were there before. So 30 years later, we were back still doing the same stuff. Anyway, this is what the sediments look like. In this case here, we have this concave up distribution of methane. And this sealed it for me. I figured, okay, we're dealing with a water column that's anoxic. There's no oxygen. This mixture of something that can oxidize methane into the system is out of the question. It's really sulfate. And this really sealed it for me. I figured that this was where we went. And the next thing that really sealed it all was the fact that we did an advection diffusion model treatment on the data from the water column, and we got this thing here. It turns out that methane was non-conservative. If it were conservative, that is not consumed by any biological process, the methane distribution would have followed the dotted line, but instead it was like this, and so we could estimate what the methane oxidation rate was. And then it became a matter of what do we say? This was, anyway, no, let's see, what I wanted to do is about 10 years later, Mary Scranton came up with this time-dependent box model. She was very concerned, very critical of us because we'd assumed steady state, and what happens in the Cariaco Basin is that the anoxic interface does increase and decrease. It used to be in those days that Woods Hole would always have a winter cruise to the Cariaco Basin and stay out of the North Atlantic, and so there was a long time series of samples there, and they could always document the location of the interface. So Mary did this. Mary's is probably more correct. We got the same result, and curiously we ended up using her models with our C14 things. John Kessler did that as part of his dissertation. So anyway, let's move on. It turns out that this is where things were sort of first proposed. You see in the literature a lot of these three authors. This paper in 1976 for the Cariaco Trench, and in 1980 for Scan Bay. Those are cited as sort of the first proposals of anaerobic oxidation of methane. Chris Martins and Bob Berner did a paper in Science, and they still sort of spelled it out as a big mystery. They came up with 4 hypotheses. They rejected 2, focused on 2. One of the ones they rejected was the answer. And that's been kind of funny. And then Barnes and Goldberg did a paper that they published in Geology, which was a brand new journal in rapid publication journal, and they worked on Santa Barbara Basin sediments and were never heard from since. It's the only work they did. Martin stayed with this for quite a while. Okay, I was thinking about this, and I put together the schematic diagram sort of to think about what to do in the future. And here you see the blue, the cyan line is the methane profile. The red line is the sulfate profile, that they kind of overlap. The hatching doesn't show up, but I'm sort of thinking this area right in here is what we call the sulfate methane transition. And so what should be happening then is as you diffuse methane up into that zone, it's going to be oxidized to CO2. So the white line is the DIC, the dissolved inorganic carbon, or the CO2 line, and it changes slope. So it means that we have basically increased the flux by adding methane in this zone. So this is kind of like a zone that everything enters and nothing escapes. Okay, and continuing on with that line of reasoning, I figured that the yellow line would represent the rate of methane oxidation, that we should have a methane oxidation rate maximum in the sulfate methane transition. And methane, it turns out, has a very light isotopic signature. So if we oxidize the methane, we're going to make the dissolved inorganic carbon pool isotopically lighter. So that should be the green line. So we basically, I pieced all this together. Well, about the same time, there were lots and lots of people working with isotopes. They were working on single species. In particular, Ian Kaplan's group, who at UCLA had two students you've probably heard of, George Claypool and Martin Goldhaber, were working with DIC, and so they were getting data that looked just like this, and they were working in places like Sandwich Inlet and Santa Barbara Basin. They were working in all the right places, and they had the profile, and I figured, well, this is an area that I'll plan on staying out of. But I wanted to say that what happened was that they were working mostly from the standpoint of the profile, the isotope profile being caused by fractionation during methanogenesis. They were thinking methanogenesis, not methane oxidation. And I should say that this was sort of considered an unknown process, and Ian Kaplan had worked with Basbecking in Australia, and of course this was an impossible process. It didn't happen. Okay, so the next one, we worry about the oxidation rate, and I want to go into that where what happened in 78, Bo Jorgensen in Denmark published this landmark series of 3 papers where he used sulfur-35 sulfate, the radioactive isotope of sulfur, and measured the sulfate reduction rate. And so basically he would add sulfur-35, recover sulfur-35 sulfide, and this stuff is really hot, it's carrier-free, it has a short half-life, and it turns out that he did this, and he was welcomed with open arms. The microbiological community, the microbial ecology community, their feeling was, at last we can measure this process, we can measure a rate, and great for this. And so he did a very comprehensive job of it. And as far as the methane goes, at about the same time, there were a few studies. John Rudd and Bob Hamilton were working on methane in the experimental lakes area, and that was, I kept puzzling over, could we do this? How would this work? One key thing about Jorgensen's study was that he convinced me that you don't have to homogenize the tracer. And let me go into this a little bit. Up to this point, I had worked with people doing primary production on ships, and there you take the sample, you add the tracer, and you homogenize it by shaking it. And I didn't see how in the world we could homogenize mud and get the methane distributed through it. And so this basically, his argument that you basically, all you have to do is not lose tracer from the system you're working on was the deal. So I figured it was worth a try. So what we did then was to make these measurements on segmented cores from Scan Bay, and we're thrilled to get these results here that showed the predicted maximum in oxidation rate, or at least I predicted that. And about this time, about a slide or so before that, I ran into incredible controversy because I managed to really touch a nerve with the microbiologists. They, first of all, seemed to think that I had, that how could I say this with models? They were very concerned about how this could happen. The second thing that they were concerned with is they said I needed to isolate an organism. And the third thing I really got a chuckle out of was they said, "You're not a microbiologist. You can't be in this area." And in truth, I've never had a course in microbiology, so I thought that always sort of tickled me. Anyway, so we started off, and we realized that if we were going to do any modeling, we had to have more resolution, more samples. And so we started off. Mark Alperin joined me. We started working with these cores like this. These are cores that are perforated, and we covered the holes with electrician's tape, and then used this little syringe sampler to actually subcore the thing. So we ended up with much better resolution. By this time, we'd acquired a box core, so that allowed us to take something like a dozen cores from about a square foot area. So we managed to sort of scotch the lateral variability question, so that worked out well. And the kind of results we got from that were like these. And so this panel here, in case you can't see, is the methane concentration. The methane oxidation rate goes through this maximum, like we expect. Sulfate reduction decreases as usual, and it turns out the sulfate reduction goes through a slight maximum right there in that sulfate methane transition. So we were really thrilled with this data, and went to the microbiologists again, and were again rebuffed. And the arguments they used this time was they said that we'd been reckless, totally reckless, that we had added a tracer to, or we'd added something to an unknown system, an unknown natural system, and this was just reckless beyond belief. And they seemed to think that the controls weren't right. that we needed to do blanks. And I don't know how you do a blank with a radioactive tracer, but that was that was the things that happened. And again, it came up that we had to isolate an organism. And so I was kind of frustrated. I was kind of taken aback, but at the same time, I figured, well, let's stay with this. I'm going to convince these guys yet. So we started off, and at this point, we started to work on stable isotopes. And I told you before, I was reticent to get into stable isotopes because Kaplan's group seemed to be, a hair away from the answer, and I just didn't know quite what the deal was. So Alperin and I decided what we would do would be to try to overdetermine the system, take an approach that most of the isotope people weren't taking. And so what we tried to do then was to divide the carbon system in these sediments of Scan Bay into 5 pools. So there was methane, there was a dissolved inorganic carbon, and we knew those two would be interacting some way. So we went on ahead and measured all these others just to be convincing. The dissolved organic, the particulate inorganic, and the particulate organic. And we sold this as an internal tracer experiment. So what it amounts to is that oxidizing methane is injecting C13 labeled CO2, and so we should be able to handle it that way if we could account for everything. So we did this by taking, these are salmon cans, and we filled the cans with a 3 centimeter segment of sediment, flushed them very well with nitrogen, sealed the cans, and I got them back to the lab and used this device here to enter the cans and puncture, take out the gases, and then take the lid off, and then do all the other measurements on the cans. So this was sort of what we did. So one of the things I've always tried to do is use something that's simple and have like mason jars and salmon cans. Anyway, it turns out that what we got for results from this were the fact that the dissolved inorganic carbon concentration looked like we expected it to. And it turns out that the isotope distribution was just like the one that Kaplan and his students were measuring, so we were very pleased with that. That was the first thing. And the second thing we looked at then was the C13 of the methane and the deuterium content of the methane. And it turns out that in this case here, what happens is that after it passes the sulfate methane transition, the isotope concentration becomes heavier. So what it amounts to is that's the residual methane that didn't get oxidized and didn't get stripped of its light isotope. So we ended up with a heavier signature there. And we did a bunch of modeling. We were able to extract the fractionation factor and from that predict what the rate should be, and everything seemed to match up very well, and we were very pleased with all this. Well, this is sort of where the controversy ended, because I gave a seminar. Alperin hadn't quite finished his thesis, and I was in Germany and gave a talk at Konstanz. The professor there was a fellow named Norbert Finning. He was a great, very tall man. And so I gave this talk, and I was kind of used to this because I'd give these talks to microbiology audiences. Grad students would all come in and assemble, and they figured they were going to see a slaying or something. And I, everybody would leave puzzled. They realized that there was something they didn't quite get about this. Anyway, Finnig listened, and he stood up to sort of take questions. And before he did that, he said, just wanted to say that we haven't believed this. This has been something we've been very negative about, but this is compelling. The stable isotope information this satisfies me. We have to get to work. And you could just see jaws drop all around the audience. And then I gave another seminar about 2 days later in Bonn at my friend Hans Trooper's lab. And Trooper greeted me by saying, "What did you say to stir them up so down there?" So anyway, this was the deal. So anyway, the controversy raged for about 10 years and ended very quickly. And then it turned out that at that point we ended up with the criticisms then being, well, it can't be globally important because it only occurs in anoxic basins. And the other thing was that this just isn't a very important process. And they kept saying you still have to isolate the organism. So that was the way it went. Okay, well, let me back up here. What I did then, what I've tried to do with students is to, they're fully invested in a question and a problem. So when Alperin finished up and went to North Carolina, he carried anaerobic oxidation of methane with him. And so we, at that point, had an opportunity to get into some terrestrial work. So I did, with Steve Whalen, we did Tundra and Taiga methane flux time series. We had set up a series of stations right near the campus in the Arboretum. We did also in at Bonanza Creek, LTER. We did a pipeline haul road transect. Steve and I did that. It took 3 trips back and forth, taking samples every 20 miles and setting up stations. We also had support to work at Tulick Lake, and that's where Shannon Regley and Jennifer King joined me, where we did flux time series. We did a bunch of water table manipulations. That was a exciting period because we had pumps that were run by batteries. The batteries were powered by solar panels. And the one thing we discovered was that the ground squirrels, the 6.6, loved vinyl insulation. And we were constantly fighting shorts and dead batteries getting that work done. And then Jennifer King here did the pulse labeling, and that was a very, very intensive thing, and worked out very well. It turns out that we learned that the methane is produced very quickly right in the root zone and is emitted as methane. So we did that using C14 CO2 and saw it come back. Well, during this period, I had an opportunity to go to the Black Sea there was a big cruise right before the NOR. The original NOR was refitted, and they planned to spend about 6 weeks in the Black Sea. So I was chief scientist on the last leg because we were using radioisotopes and they wanted to keep us at bay. And so we were able to work in the one central station, and at that point there was the Soviet Union, so we were shadowed by a Russian ship the whole time. And we basically just worked at this one station right in the center of the Black Sea. And what we did in a 2-week period was measured methane. We hammered the methane profile. And we also measured methane oxidation rate using two methods. We used, Bess Ward joined us and used her C14 method. In this case, the specific activity is such that you have to add, you're increasing the amount of methane by adding the tracer. So you had to extrapolate back, do a series of concentrations and extrapolate back to in situ. And we had worked on this tritium-labeled methane. We managed to synthesize some of that. Ken Sandbeck helped with that. He had a microorganism in his back pocket. He was one of the postdocs who, an organism that could demethylate amines. And so we bought tritium-labeled methylamine and made the tritium and did it that way. And it turns out that the 2 rates, we did these parallel, they agreed within a factor of 2, which I still think was just remarkable. We were incredibly lucky to have that happen, I thought, given the stage. So basically using the reservoir, at least 2 of the R's, we used the reservoir and the rates. We were able to put together this budget for the Black Sea. And these are all, the sinks are all the things that remove methane from the Black Sea, like evasion into the atmosphere, oxidation in sediments, the outflow at the Bosporus, and water column oxidation. And it turns out that this, well below in the really truly anoxic part of the Black Sea, this is a factor of 100 larger. This is a huge sink. It was a major sink for methane in the Black Sea. And we could only account for about 13% of the needed source. We just couldn't do it. Well, one of the things that happened very quickly after that was we started to open up and learn more about the Black Sea from the Russians, and it turns out that the whole northern part of the Black Sea, remember our station was right in here, is filled with seeps, with vents. Up here, right at the mouth of the entrance to the Caspian Sea, is one of the world's largest concentrations of mud volcanoes, which are driven by gas emissions, methane emissions. The whole thing was covered this way, so we realized that, we had been incredibly lucky because we've been studying this as a diffusion control process in sediments, and we can model and do all these nice measurements on mud and all this kind of stuff, but it turns out that really in the real world, we have a situation that looks like this. So we had been living in this little paradise down here where things were under diffusion control, but then we get up to these larger features like seeps that are bubbling, and then we have these mud volcanoes, which are enormous cataclysmic eruptive events, and we don't have a way to quantify them. So down here we could model, we could do all the isotope work and do all this stuff, but when you get out here, these all originate from deeper in the earth, and the fluxes vary they're 1,000 or more times larger. So these were the things, particularly in here, that we thought were driving the Black Sea. So we managed to tag on another cruise and went along to collect more samples. We collected one set of samples for C13 analysis, and when I came to Irvine, they were oxidized, and when Stan Tyler first got his mass spectrometers running, he ran those samples for us. So we had one point, and then we went on ahead and really didn't know quite what to do. We were on this cruise, we were going to take more samples, and one of the things that happened was sort of in a casual lab conversation, Dave Valentine mentioned, we just were setting up the AMS here, and he said, I wonder what C14 would look like. So I checked quickly with Sue, and it turned out that we could use 20-liter samples, and it looked like we could measure C14 of methane in the Black Sea. So that became another project, and that's what John Kessler worked on. So going on to the next thing, this shows the C14 concentration of methane in the Black Sea, and it turns out that the average here is about 13% modern carbon. So that means that there's a mixture of carbon in the methane. Part of it must be modern, part of it is fossil. It's radiocarbon dead. It has a 0 PMC, and using that time-dependent box model, we were able to show that John did this, and it turns out that these two things here, the methane profiles 13 years before and after, and it turns out that these are the estimated fluxes by depth interval to the Black Sea, what it would take to maintain that 13% modern carbon. And we were able to, when John was doing his dissertation, we got a set of samples from the Germans who'd collected seep gas right at the seep using a submersible. And John came back and analyzed those samples, and they're still modern. They have 5% modern carbon. We still don't understand that. We don't know where it comes from. Went to the Carriacou Basin, as I said, 30 years after the first trip, and learned that the water column was only 2% modern. And none of us had ever considered seeps in the Carriacou Basin. We probably should have, but we never thought of it. It turns out that in the sediments, everything is modern. So we have two systems. We have particles that are fixed in the upper part of the Carriacou Basin are falling into the sediments. And so they're much more modern carbon than the water column. So the water column seems to be driven by seeps, by fossil methane coming into the Carriacou Basin. This shows a similar sort of slide. What's happened in the Carriacou Basin is that this is the data that we got the first time 30 years before. This is the data we got on the cruise John and I went on. These are model predictions of what will happen to C14 of methane, or actually to the methane concentration by the year 2065. Wasn't that it? And then this shows basically the distributions, the locations in the water column where the methane is being injected by seeps. And interesting thing about this is that John took this model, instead of running it to steady state, he ran it backwards and he ran it forwards. So running it backwards, it turns out that basically sometime, when was it, 1965 was it, that there were two of the largest earthquakes the area has seen. And so those must have shuffled or rearranged the plumbing and allowed the methane flux to increase. And if things stay the same, we don't have another earthquake. This is what the predicted profile of methane will look like in the Carriacou Basin. The stable isotopes we ended up using, they basically sort of fell out of the data, out of the samples naturally. So for the Black Sea, we have basically this first set of del D measurements. Those were, well, I'm sorry, the del D measurements, we didn't know how to measure del D at the time, so we had a lot of scatter. But we did measure del C13, and you can't distinguish the points, but this is a merged profile. It shows our del C13 from 13 years earlier and the current ones. And it turns out that we used that to establish steady state. This is one of the things that really, late in my career, learned that steady state matters and that it was one way to do it. But basically, we established that things were in steady state. And so this is what the profiles for isotopes of methane look like in the Black Sea. And we go to the Carriacou Basin, and it turns out that they're almost mirror images. Let me show you this once more. Questions, why? What's going on? Why do we have something as fundamental as the end of the line as methane behaving so differently in two anoxic basins, which are unusual in themselves? We were really puzzled about that. And what happened was that we learned that the Black Sea, using that del C13 profile, we could establish steady state. The concentration may have differed, but the isotopes were spot on, and so we could establish steady state. And we ended up realizing that it was caused by anaerobic oxidation of methane under open system, steady state conditions. In the case of the Carriacou Basin, the reason those profiles are so different is that it's non-steady state, You're still adding methane. The methane's increasing in the water column. It's not all oxidized, and so it turns out that it basically stays lighter, isotopically lighter. But from both of these, you could go through and calculate the fractionation factors, and they agreed perfectly with the ones that Alfred did in sediments of Scan Bay many years before, so we were quite pleased with that. The last thing I wanted to do or get into is global importance, and let me move on ahead with that. First thing I wanted to point out is how widespread, well, just the earliest studies were done in diffusion-controlled sediments. I've already told you that, and this allowed us to do modeling, to make rate measurements, to do stable isotope studies, and sort of deal with things as a kind of closed system. And the thing I wanted to point out is that there was extensive replication. The very beginning of all this, it turned out that every time you made a measurement, 2 years later, somebody had one there. So with all the concentration profiles, the rate measurements, some of the isotope work, the rate, all these things were replicated by people like Al Duvall at Washington and Chris Martins. We were really, it was a really very, very hot item. But then we realized that these more recent things that we're dealing with seeps, these violent, relatively violent sort of things happening. And there's no rate measurements, but they can use biomarkers. The CSIA stands for Compound Specific Isotope Analysis. So this is where you isolate a compound chromatographically and analyze its isotope concentration or isotope ratios. FISH is an acronym for a genomic technique called fluorescent in situ hybridization. And all these things have sort of documented the occurrence of, but not the rate of, anaerobic oxidation. It turns out that FISH has almost become the golden standard for identifying anaerobic oxidation. And one of the things I wanted to point out is that AOM, anaerobic oxidation, is a stealth process that operates in a subsurface zone. Unless you're looking for it, you don't see it. You see the effects, but you don't realize what you're seeing. And it turns out that this is a huge sink for methane in the ocean. And it turns out that the only reason the ocean is a small methane source to the atmosphere is because of methane that leaks out of fecal pellets in the mix zone. And so this is what basically drives the ocean as a source. It turns out that this starts off, so here's the initial work in Chesapeake Bay. I want to just step through these and show where the studies appeared. This one is for the 1970s, and so at that point we have Chris Martins. We've gone to the Carriacou Basin. Bo Jorgensen, Henry Blackburn, Niels Iversen began to work in Denmark. Ross Barnes in the Carriacou Basin. It turns out that Al Duvall and Saanich Inlet, and we had established our work out at Scan Bay. The next one then shows still more things. So this basically, we've got more studies in Saanich Inlet, more Carriacou Basin. Ron Ormland started to work on lakes, Big Soda Lake and Mono Lake in California. And going on here, we start to get into the 1990s. This is where we end up with studies of the Hawken-Mosby mud volcano off the Namibian coast, the Argentine basin, more work in the Amazon basin. Bob Allard did some work there. It's just amazing to see how this all grows. And then the last one, the 2000s, it turns out that there have been two deep sea drilling project legs that have emphasized this. More work on the Hawken-Mosby. And we've been to Scan Bay. I think Valentine has been twice or three times. 3 times. John's been twice. So anyway, we were in Scan Bay a total of 9 times on this project. And so this is really quite, or this problem. So anyway, this sort of shows how it's grown. And what I wanted to do then is to show how important it is. And it turns out that when I started to think more broader than Scan Bay, I figured that conservatively all I could do was to really worry about high organic shells. And so that basically, and it turns out the curious thing, since it's a diffusion-driven process, it's pretty much the same. If you integrate the oxidation rate with depth, it's pretty much the same thing. So it became a kind of simple calculation. I conservatively estimated that 70 teragrams of methane were consumed each year by anaerobic oxidation. Well, by 2000, it turned out that Kai Henrichs and Antje Boyches, both from Germany, they had advantage of that all the spots on the map, the last map you saw, and they broke the ocean up into zones and basically went, considering inner shelf all the way to the lower margin, They total something like 304 teragrams per year. And they also threw in the possibility that seeps could be adding about another 80. And so we're talking close to 400 teragrams, and I think it's probably still an underestimate. It's huge, but and that is what keeps the ocean from being a methane source to the atmosphere. Another study that was done, this was part one of the deep sea drilling project or ocean drilling program studies by Stephen DeHaan, and this was a paper that was published in Science, two papers in Science. And one of these, what he'd done was to take open ocean and continental shelf studies, and of course you're looking at segments of several hundred meters in sediments and very, very small rates. And he modeled and measured all these things and came up with a global estimate of 10 to the fourth. I think that's outlandish, but it's a huge number. And it turns out that there are these, I'll get into this in a minute, there are these methane oxidizing communities. It turns out if we look at Cariaco Basin and Black Sea with using the C14 corrected everything, it turns out that they're still fairly small numbers, and this evasion of the atmosphere seems to be settling in at around less than 20 teragrams per year. So let me see, I'm going to break here. How am I doing on time? I better switch. Okay, so now what I'm going to do is, it's incumbent on somebody giving a talk like this to talk about the future. So what's next? It's not all done, I can assure you. One of the things that's, I think, looming is a very important question is this business about methane clathrate hydrates or hydrates. DOE is looking very hard at trying to exploit these as an energy source. We know that they dissociate naturally, and so the real question is how much are they adding and how much actually gets into the ocean? That's one of the big questions. So we have to some way quantify the clathrate contribution to the global budget. This business of the subsurface maximum in the Oxic Ocean has been studied extensively by Dave Carl, and it turns out that it has lots of potential still. They've measured rates. They have a good understanding. What we need is a really careful study there using radon and measuring gas exchange as well as oxidation rates and see, and we have the capability to do all that. I think that would be a really great project. One of the things we run into is in most of the ocean, the concentration of methane is nanomolar. It's millimolar in the Black Sea and the Cariaco Basin, but it's nanomolar in the ocean, yet it's being oxidized. How? This is an incredible kinetic question, so that's going to be one of the big things in the future. This whole business of quantifying the vent and seep contributions and also mud volcanoes, there are lots of papers written about it and lots of very authoritative estimates, but they're guesses. Every one of them is a guess, and so we need to find some way to quantify that, and I think that C14 will play a key role there. One of the things that we've sort of come to realize is that we need to measure C14 methane in the open ocean, and with those nanomolar concentrations, this is going to take 2,000 to 4,000 liter samples and extracting all the methane, so this becomes a real big job, and so I think that's probably going to be something that's pursued by probably Mary and John. And the last question is, what is the organism and what is the mechanism? I didn't have a chance to go into too much about the mechanism, but I have to tell you, after being advised time and time again, you have to isolate the organism. It still hasn't been done, and and so I have to kind of chuckle about that. So that was one of the original questions. And let's see here. Let me go through and this give you some idea. This is a couple of pictures from Ken Janda's webpage. We've had a clathrate project together. This is the type 1, the type that forms methane. The methane forms, and what you have is these lattices with a water molecule at each of the apices, and they have two 6-sided faces and 12 5-sided faces. So this is the type 1. This one here shows the chlorine hydrate. This is also from Ken's webpage, and the interesting thing is that this was the first hydrate synthesized. It was done in 1811 by Humphrey Davy. So this has been around a long time. Okay, this shows a shot from the Malik well. This was a well that was drilled into a hydrate prospect in the Mackenzie Delta in northern Canada. And this is the first successful production test for methane from a clathrate hydrate. And let's see, this shows the open ocean subsurface maximum in both the Atlantic and the Pacific Ocean. It turns out that these were early on. I mean, this as soon as people started measuring gases in the ocean using gas chromatography, these were established. And it turns out that modeling work has shown that this has got to be conditioned. It's a source, a local source, probably leakage from fecal pellets, copepod fecal pellets, and then it's oxidized as it diffuses into the atmosphere. And so understanding that is I think a big priority. This shows a section in the Pacific Ocean, and you can see concentrations are down to 0.5, 0.6 nanomolar. And basically, how do they get that way? So one of the questions that we often run into is with this business of methane sinks, I've emphasized mostly the anaerobic, but we know in the open ocean there's got to be aerobic oxidation occurring. And a couple of the things that are unique about these is the fact that if you oxidize methane aerobically, you add CO2 that decreases the pH and can dissolve carbonates. But in the case of anaerobic oxidation, you're producing bicarbonate and you're also producing sulfide, and those increase the alkalinity. And so one of the key tip-offs early on, in fact, geologists were my biggest supporters because we finally found a mechanism to explain the light carbonate cements they were finding. So that was very, very interesting. So anyway, looking at this shows a chimney that grows on the shelf in the Black Sea. This was a measurement these pictures were taken from a submersible, German submersible, and it turns out that these things are just loaded with methane bubbles bubbling through. And it turns out that they're kind of soft. They're 300 or 400 meters, so they're well below wave action. And it turns out they knocked one over, and typically it's this sort of pinkish, fleshy, methane oxidizing bacteria. And so they measured rates and found that this all fits in. I think Walter Michaelis in the science paper basically said that he thought that this was probably what life on Earth looked like before oxygen entered the atmosphere. So look at it for that. Another thing that's happened is that off the spreading center axes in the ocean, they've discovered these warm vents. And this is one was called the Lost City hydrothermal field. And it turns out that there's warm water that apparently reacts with rocks to make methane. This is called the serpentinization reaction. And it turns out that this stuff, basically these towers are made of brucite. It's a magnesium carbonate mineral, or magnesium hydroxide carbonate. And the water is pouring out of these things, and it turns out that there's methane production, methane oxidation, all kinds of things going on. And these things are 70 meters tall. They're at the bottom of the ocean, but these things they call it a lost city. And finally, this business about the communities living around where methane is being oxidized, it turns out they aren't living on methane. What they're doing is living on sulfide that's produced when methane is oxidized anaerobically. And Tina Troida and a group of people in Bremen were the ones who really came up with this. And so they have shown that these communities are conditioned by how near the surface this sulfide boundary is. So if it intersects the surface, you end up with a slimy filamentous bacteria called Beggiatoa that grows in profusion. So that's easy to see from a submersible. When it's a little lower, there are these clams called Calyptogena. And then you can go to the acarics further down. It turns out that what the Calyptogena clams do is that they stick their foot into the sulfide and they operate on the energy difference between oxidizing sulfide and oxygen. They can take in oxygen through their gills, and basically they operate this way. So they're living on sulfide oxidation. And let's see. Here we go, acknowledgements. So I've all through the talk tried to pepper and show who did what, where, and when. Our work has all been supported by NSF, by a big variety of programs at NSF, ocean sciences, polar programs, molecular and cellular biology. I never thought that would happen, but it did because of this Lexin, the Life in Extreme Environments program we did with Dave Valentine. We've also had some support from NASA and EPA. And the Keck Foundation provided the AMS as well as mass spectrometer facilities here. People who've been involved are former students. There's David Heggie, who did a master's with me. He's in Australia now. Mark Alperin did really yeoman work up in Scan Bay with us. Steve Whalen went to the Black Sea the first time and helped set up the transects of the time series transects and so forth in Fairbanks. Shannon Regley was here. She decided that she really wanted to be a teacher, and so she's become the kind of science teacher that you all wish your kids could have or your grandkids could have. Jennifer King, who's right here, worked on pulse labeling. Dave Valentine came and worked on hydrogen. I didn't have a chance to get into mechanisms, but he's really at the forefront of what mechanism pulls this off and makes it work. He's able to do careful thermodynamic calculations and show very clearly what works and what doesn't work. John Kessler did the C14 work, and Mary Pack is the current student, and she's working with Sue Trumbore and me using tiny concentrations of C14 tracer to measure methane oxidation rate in the open ocean. So she is basically, we found that it's so difficult to work with radioactive materials at sea that we just decided that let's try this, and it looks like it works. Mary just returned from a month at sea. She spent Christmas on the bounding main, and everybody on the cruise said she did great, so that's the deal. And I guess finally what I'd like to do is just say that I really want to stay in touch. I won't be here, but I do want to stay in touch. UCI's been good. I have feel a sort of special kinship with the people who are hired on my watch. I see a few of them around, people like Francois, Jay Familietti, Jin Yi, Keith Moore. And then I was going to tell a special story about the three that were hired. This is the real coup. We had half a position that we were going to split with E&E biology. And so the search committee worked on this, and they came up with two people that they couldn't decide between. So we went to the dean, went to Ron Stern and said, what do we do? And he says, oh, I'll talk to their dean. We'll add another half. And so we ended up adding a half. And so we ended up making two offers. We made an offer to Diane Pataki. We made an offer to Kathleen Treseder. They both accepted. Jim Randerson came as a spousal hire. And out of this initial half position, we have 3 faculty members, 2 who have won the McElwain Award. And couldn't be better. That's something I smile about every day. So anyway, I think that does it. If there are any questions, I'd be happy to answer anything. But thank you all for coming. Thanks a lot.
Again, Bill, it's been wonderful. We'll miss you. We'll miss you. Thank you.
And I'm a new recent hire on. Okay. I met Bill Reeburgh at Woods Hole Oceanographic Institution when I was an assistant scientist who's on staff from the University of Alaska. He appeared at the chemistry department office and wanted to visit some labs. Since he had measured C14 in his lab, natural, he was interested in seeing our setup. So he regaled me with stories of how he measured C14 using counting techniques too, and even sent us some of his prized oxalic acid later on. Bill was faculty hire number 4, I think, after Ralph Cicero, Sue Trumbore, and Darren Tilley visiting the University of Colorado. We would frequently have faculty meetings in Bill's office. Just the 6 of us sitting around picking up names for the departments. We were originally the program in geosciences. During our discussions, we politely argued as to whether system or science should be plural or singular. This went on for months. Ralph and Michael finally won because they really felt that they should be singular. But it was Bill who led the charge, toiling long hours to craft the proposal for establishment of department status, which we attained in 1994, and then the doctoral degree program a year later. This was record time, as Sue explained. for establishing a graduate degree in the UC system, which involves system-wide review at the University of California President's Office, among many other steps. And its success was mostly due to Bill Reeburgh's thoroughness and his communicative skills with the administration. Remember Carol Sokol? They got it through. Yeah. Bill is a communicator. You all know what I mean. He likes to have face-to-face conversations rather than use email. Here's what I mean. He will appear in my office door and ask how I'm doing. Then he will ask me a question or tell me a bit of information, like the latest news on EU politics. I will respond. Then he will tell me that it reminds him of an experience he had while in Alaska. Learning at the end, kind of like a moral to the story. Then, as quickly as he appeared, he will say goodbye and disappear down the hall. We will miss you, Bill. And to remind you of us, we've crafted something. We know how much you like handmade items, given your skills with woodworking and leatherworking. So we signed a plate in honor of the occasion of your retirement. It is color-coded according to the area of our research. Or personal color choices. Green and brown for land, blue for ocean and atmosphere, red for fire, there's only 2 of those, and clear and gray for ice. Yeah, I know ice is not really gray, but that's colored glass. It has some of those copper wire flowers that you like in the plate in my office, so we hope you like it. Thank you, Bill. We appreciate you because you are a good colleague, always thoughtful, willing to give people the benefit of the doubt, and put in the time for common good, that is for system science. We hope you will not be a stranger in Minnesota.
But I thought Ellen has those pictures hanging outside her office of how the department grew and changed over the years. So we made a little poster with those photos, and it's over by some of the food. So you can take a look and take it with you when you go.
Yeah. Good about Nancy.